Welcome, science friends, to point mutations. So in the previous video, we talked about different types of mutations, and we're going to focus on substitution mutations, which are a point mutation. Point means we're looking at a single nucleotide. So there are a few different types of substitution mutations. First is a silent mutation. When a substitution happens and nothing changes the amino acid that's being produced, it is considered silent because nothing in the protein is changing. This is where all of those repeats and redundancies of our amino acids, when we look at our codons, there are 6 different ways to make that amino acid. So if there are changes that are one of those 6 codons, our protein will still come out exactly the same. So it's considered silent because we're not seeing a change in the protein even though we see a change in the code. In this example, we are changing a C to a U on that second to last codon. So instead of a GGC that codes for glycine, we get a GGU. But luckily, that still codes for the glycine. Again, this has no effect on the protein. We're still getting the amino acid we need, so it's considered silent because it's not noticed in the end product. The next type of substitution is called a missense. So this happens when a substitution changes the amino acid that's being produced. This can affect our protein. If our protein doesn't have the right amino acids in the right order, that can change how the protein functions. So in this example, again, we're working with that second to last codon. The wild type is a GGC. But when we make a missense point mutation and we change that first G to an A, instead of GGC, we get AGC. And when we change to AGC, we're producing a different amino acid than the wild type. The wild type, the GGC, codes for a glycine, which is the correct amino acid. But in this missense point mutation, the AGC codes for a serine, and that will change the amino acids in our protein, thus changing our protein and its function. This could have a really big, dramatic effect, like we saw with hemoglobin. We went from the round to the sickle shape, but sometimes it doesn't matter as much. Proteins are huge. They are hundreds of amino acids long, and sometimes it's not a meaningful change, but it depends on the protein that's being made. The last type of point mutation is called a nonsense mutation. These are the worst kinds of point mutations. A nonsense mutation is when a substitution happens from a wild type to a stop codon, and that stop codon comes early. So in our example here, we're looking at this second codon. In the wild type, we have AAG, which codes for lysine. But in our nonsense mutation, we swap out that first A with a U. Instead of AAG coding for lysine, we get a UAG coding for a stop. And if we stop too early, all of those other amino acids are not going to join the protein. Now our protein is going to be too short, and that's really bad. If our protein is too short, it gets stopped early, it definitely cannot do its job. Of the 3, nonsense is the most dramatic and devastating to the end protein. So just a quick review. The very top in the white is our wild type. That's what our gene should look like. Right underneath that is our silent mutation. We substituted out a nucleotide, but it had no effect on the amino acid being made. Therefore, no effect on our protein. That redundancy in our code, there are many different ways we can make glycine, is our friend. It's helping us out. The missense is the second point mutation we talked about. That means we're swapping out one nucleotide for a different nucleotide that has a change in the amino acid being made. So here, instead of a glycine being made, we swap out the G for an A, and now we're making a serine. Very different amino acid, could potentially harm our protein. The last one is the nonsense. This is the worst one. Instead of just nothing happening like a silent or changing the type of amino acid in missense, it's a premature or early stop. And if we stop too soon, our protein won't be long enough, and therefore it's very likely to not work properly. Again, sickle cell anemia is a great real-world example because a change in just one nucleotide can change how this protein functions and even change how the cell looks and functions. We see sickle cell anemia mostly in Africans and those of African descent. 1 out of every 400 African Americans are affected by this today. That was Point Mutations. I hope it helped.